Good afternoon, I'm Singha.
I'm Anneke.
And I'm Aija.
And today we're going to talk to you about this guy, the CRISPR-Cas9 system, and how it can improve gene editing therapeutics in SCID patients. The immune system is a complicated web of interacting cells and compounds that provides us with the capability to recognize and fight potential pathogens, essentially protecting us from microbial invasion. One of its two main branches is the adaptive arm, consisting of T and B lymphocytes, key fighters in this war against infection. They begin their cellular life as hematopoietic stem cells, or HSCs, before undergoing a series of differentiation steps, tightly controlled through signal transduction and gene regulation. Signals are received from other immune cells via messenger molecules called cytokines, which interact with the receptors on the lymphocytes to initiate their next life stage, to be or not to be a B or T cell. Following this complex cascade of events, the final cells in these intricate pathways are mature lymphocytes, ready to attack. Now this is all well and good, but what happens when this sophisticated system breaks down?
Severe combined immunodeficiency disorder is a rare syndrome that arises from genetic mutations in these very pathways. This leaves the 1 in 58,000 born with the condition unable to fight off infection as they're severely immunocompromised. Microbes that are usually harmless to us pose a serious threat to those afflicted, with untreated children often not surviving their first year of life. One of the many forms of SCID is caused by a monogenic mutation in the gene encoding Janus kinase 3, or JAK3. This kinase is functionally coupled to a large range of cytokine receptors, including the growth factor receptor on undifferentiated T cell progenitors. The mutation in this important kinase manifests as a complete block in T cell maturation, with young T cells unable to translate cytokine signals to a mode of action. The subsequent lack of a mature T cell population has vast knock-on effects, including a defective B cell population due to the loss of T to B cell signaling. This single gene mutation therefore leaves JAK3 deficient patients completely stripped of a functioning adaptive immune system.
There is currently only one established treatment for this disorder, which involves transplanting HSCs from the bone marrow of a healthy donor into the patient. This transplant is undertaken in the hopes that the donated stem cells will rebuild the patient's immune system. While the treatment has been successful, as with any transplant, there are risks. Of particular concern is graft versus host disease, or GVHD, in which the donated cells recognize the host tissue as foreign and proceed to attack it. This manifests as a range of symptoms, from rashes and diarrhea to liver damage and possibly even death. While HLA matching can prevent GVHD, a lack of suitable donors and the severity of untreated SCID often leads to less than ideally matched transplants going ahead anyway. Due to the limitations of this approach, researchers focused on using gene therapy to fix the underlying cause of the disease, the single point mutation in the DNA. Gene therapy is an experimental technique with the ultimate goal being to introduce genetic material into cells to compensate for or to correct a disease-causing gene.
Previous SCID gene therapy trials used viral vectors in order to integrate a healthy sequence along with the associated promoters into the patient's own HSCs ex vivo. The transduced cells were then reintroduced into the patient and expression could occur, leading to a healthy T cell population. While this did see some successes, severe adverse effects were also observed, with several patients developing leukemia. This was a direct effect of off-target gene integration due to this therapy's lack of insertional specificity.
So here we pose to you the two main problems with treating JAK3 deficient SCID. If these patients require functioning T cells, yet transplants can lead to graft versus host disease, and viral vectors pose the risks of off-target mutagenesis and cancer induction, what else can we do? A recent study has provided us with an answer, a futuristic model of gene correction therapy that combines the groundbreaking genome editing technology with the relatively new knowledge that mature cells can be reprogrammed to de-differentiate into primitive stem cells.
Chang and colleagues isolated skin cells from a 4-month-old JAK3 deficient patient, inducing pluripotent stem cells, or iPSCs. These can differentiate into any mature cell type, including T cells. The advantage of using these cells in therapy is that they are derived from the patient's own tissue, therefore removing the risk of GVHD when transplanted back into the patient. However, we still have another problem to deal with. These iPSCs still carry the mutation in the JAK3 gene.
Enter CRISPR-Cas9, a revolutionary new gene editing technique that has taken the scientific community by storm. Unlike previous editing technologies, the CRISPR-Cas9 system can easily be reprogrammed to target any genetic sequence within any genome, guided by a short sequence of engineered RNA called guide RNA. Once there, the wild-type Cas9 nuclease induces a double-stranded break in the DNA, providing a window of opportunity for genetic modification to occur. While this is an exceptionally specific process, the opportunity for off-target mutagenesis is still a possibility.
To address this concern, scientists engineered the Cas9 nickase, which induces a single-stranded nick as opposed to a double-stranded break, ultimately requiring two closely juxtaposed nicks in order to trigger the same genomic disruption as the wild-type Cas9 nuclease derivative. This effectively boosts the recognition sequence from 20 to 40 nucleotides, as it requires two guide RNAs, one for each strand, drastically reducing the chances of these off-target effects. This extreme specificity overcomes many of the issues associated with viral vector gene therapy. So how did Chang and his team apply this to their study?
In order to correct the single point mutation in the patient's JAK3 locus, in this case found in exon 14, the team first had to synthesize a series of guide RNA molecules specifically targeting introns up and downstream of the mutation. The researchers then constructed plasmids, expressing either Cas9 or the modified Cas9 nickase, along with a single guide RNA or paired guide RNAs, respectively. Now that the cutting machinery is ready to go, what happens at these break sites?
The Cas9-induced DNA breaks will trigger the cell's own endogenous repair mechanisms. One of these, homology-directed repair, or HDR, uses a homologous template in order to restore the sequence. The team thus generated a JAK3 correctional template for HDR. This included the wild-type JAK3 gene, as well as a selectable marker, the PGK-NEO cassette, which allows for the screening of successful recombinants. All of this was loaded into a plasmid. To finally put it all together, the iPSCs were nuclear affected with the CRISPR-Cas9 and the HDR template plasmids.
After 2 weeks of growth, iPSCs were then selected, expanded, and the efficiency of CRISPR-Cas9 gene correction was quantified through RT-PCR and Western blot. Restored gene expression was demonstrated in 100% of iPSCs nuclear affected with the Cas9 nickase and paired 1 and 2 guide RNAs.
Furthermore, whole genome sequencing reveals no off-target mutations in any of the 1,450 potential off-target sites, suggesting strong specificity for CRISPR-Cas9-directed gene editing.
Correction of this mutation also restored the differentiation potential of early T-cell progenitors in vitro, which can lead to healthy T-cell populations.
The generation of patient-specific gene-corrected iPSCs overcomes many of the issues associated with previous treatments, such as graft versus host disease and off-target oncogenesis. In theory, these JAK3-corrected patient-derived iPSCs could be differentiated out and transplanted back into the patient, reconstituting their T-cell population and effectively rebuilding their adaptive immune system.
While phase 1 clinical trials have yet to be undertaken, these results provide a strong foundation for gene correction therapy in humans using the CRISPR-Cas9 system. It is not too far-reaching to say that we are well on the way to a cure for SCID.
While this in itself is an amazing achievement, the application of these combined technologies do not stop at JAK3. Research into the application of these techniques for the treatment of other monogenic disorders, such as certain types of muscular dystrophy or sickle cell disease, is currently being undertaken with very positive results.
Although we still have a long path to walk towards genetic disease annihilation, the combined forces of CRISPR-Cas9 and induced pluripotent stem cells have catapulted us along. The journey has well and truly begun.